Good morning friend. In this video we are talking about enzyme part 2. Before starting video I humbly request you to use earphone during watching video, and also the PDF run beside the video is given in the link of PDF is given in description box, and the link of previous videos PDF given in the description box. Now without wasting much time, let's start the video. We cover two topics: regulation of enzyme and therapeutic and diagnostic application of enzyme and isoenzyme. Fourth topic is regulation of enzyme. In regulation of enzyme, the enzyme activity is regulated by 6 ways: allosteric regulation, activation of latent enzyme, third is compartmentation of metabolic pathway, fourth is control of enzyme synthesis, and fifth is enzyme degradation, and sixth is isoenzyme. We discuss each of them in details. Now first is allosteric regulation or inhibition. For this type of enzyme, specific allosteric sites are present. Allosteric species are additional site other than active sites for enzyme. This allosteric modulator bind to allosteric sites and regulate the enzyme mechanism. There are two type of allosteric binder: positive allosteric binder and negative allosteric binder. Now when positive allosteric effector binds to allosteric site, it increases the enzyme activity and known as activator site. Example of a positive allosteric binder is ADP, AMP. ADP means adenosine diphosphate and adenosine monophosphate. This will act as a allosteric activator. And second is negative allosteric effector bind to the allosteric site called as inhibitor sites and inhibit the enzyme activity. Example is phosphofructokinase, and second is ATP, act as a allosteric inhibitor. This allosteric inhibition is controlled by negative feedback regulation, means increase in the level of end product will result in decrease in the synthesis means जैसे जैसे end product की level बढ़ेगी वैसे वैसे synthesis कम होगा. Second way is activation of latent enzyme. Some enzyme remain inactive up to the site, but it get activated at the site of action by breakdown of two or one or more peptide bonds. Example is chymotrypsin, pepsinogen and plasminogen. Third point is some enzyme are interconvertible, means it converted into inactive to active form or vice versa. Example is glycogen phosphopyrolase, and second example is pyrolase B. The third factor which affect the enzyme regulation is compartmentation of metabolic pathway. Now the metabolic pathway include two pathway: catabolic pathway means breakdown and anabolic pathway means synthesis, are occur differently in cellular organism. Take one example that fatty acid synthesis takes place in cytosol, whereas the fatty acid oxidation means breakdown takes place in the mitochondria. Now in this table there are different metabolic pathway given which are occur in which organelles are given in the table. First organelle is cytoplasm. In cytoplasm, fatty acid synthesis takes place and purine pyrimidine catabolism means breakdown takes place. In cytoplasm, aminotransferase, peptidase, glycolysis and HMP shunt occur. Second organelle is mitochondria. In mitochondria, fatty acid and amino acid oxidation takes place and metabolic pathways are Krebs cycle, urea synthesis, electron transport chain and oxidative phosphorylation occur. Third organelle is nucleus. In nucleus, biosynthesis of DNA and RNA takes place. Fourth is endoplasmic reticulum. In endoplasmic reticulum, protein biosynthesis, triacylglycerol and phospholipid synthesis and steroid synthesis takes place, and reduction of cytochrome P450 and esterase takes place. And the other organelles and their enzyme and metabolic pathway are given in the table. The fourth factor which help in enzyme regulation is control of enzyme synthesis. Most of the enzyme are rate limited. They are present in very low concentration, and the amount of enzyme is control the velocity of the reaction. Most of the enzyme are rate limiting enzyme and have short half-life and hence efficiently regulate the enzyme level. There are two type of enzyme: constitutive enzyme and adaptive enzyme. In constitutive enzyme, the level of enzyme is not changed and remain fairly constant. And second is adaptive enzyme. In adaptive enzyme, the concentration increases or decreases as per body need. Now we are discuss induction and repression. Inductions means increase in the synthesis of enzyme, and repression means decrease in the synthesis of enzyme. This induction and repression determine the enzyme concentration at gene level. The fifth factor which help in enzyme regulation is enzyme degradation. Each enzyme have their self-destructive capability, but it vary according to the enzyme. In general, the key and regulatory enzyme are most rapidly degrade. Some enzyme and their self-destructive time are given in the following table. That is, LDH4 have degradation time of 5 to 6 days. Second is LDH1 has self-destructive time 8 to 12 hours, and amylase has 3 to 5 hours for self-degradation. The sixth factor which help in enzyme regulation is isoenzyme. The definition of isoenzyme is when same reaction is catalyzed by two or more different molecular form of enzyme, then it is known as isoenzyme. This different enzyme has generally different kinetic and regulatory properties. Example of isoenzyme are hexokinase 4 and LDH. Now fifth topic is therapeutic application of enzyme. Some enzyme are used in the treatment of some diseases are as follows. First enzyme is bacterial aspergillus used in the treatment of leukemia. Second enzyme is chymotrypsin used in the treatment of inflammation and edema. Third enzyme is collagenase used in the removal of dermal ulcer and several burns. Fourth enzyme is halodeurinase which promote a rapid absorption of drug injected subcutaneously. The fifth enzyme is lysozyme used in the treatment of infection of eye. Sixth enzyme is penicillinase used in the treatment of person who is allergic to the penicillin. Seventh enzyme is renin used in the treatment of gastric achilia. Eighth enzyme is streptokinase used in the treatment of myocardial infarction to dissolve the clot. The ninth group of enzyme like pepsin, trypsin, peptidase, lipase, amylase, esterase and cellulase used in the treatment of GIT disorder and chronic pancreatitis. Now we are studying the diagnostic application of enzyme. For enzyme activity, the biological fluid is important. The enzyme are divided into two groups, plasma specific or plasma functional enzyme, and second is non-plasma specific or plasma non-functional enzyme. Now first part is plasma specific enzyme. These enzymes are generally present in plasma, means present in more concentration in plasma and in less concentration in tissue. They are mostly synthesized in liver. Example is lipoprotein, lipase, plasmin and thrombin. Improper function of liver or genetic disorder leads to the deficiency of enzyme. Deficiency of ceruloplasmin leads to the Wilson disease. The second type of enzyme is non-plasma specific or plasma non-functional enzyme. It is mostly present in tissue. The activity of this enzyme is used to diagnosis and prognosis of the disease, means it is a marker of the disease. Example is digestive enzyme. The digestive enzyme are amylase, pepsin, trypsin, lipase are present in a plasma and it is also known as secretory enzyme. Increase in the level of this enzyme indicates cellular damage or increase in the rate of turnover or proliferation of the cell or increase in the synthesis of cell. And hence this enzyme is used as a marker to detect the cellular damage. Now in this table we are discuss the following enzyme and their which enzyme diagnose the disease. The first enzyme is amylase, which is diagnoses the acute pancreatitis. Second enzyme is serum glutamate pyruvate transaminase, used to diagnoses the liver disease, that is hepatitis. Third enzyme is serum glutamate oxaloacetate transaminase, used to diagnoses the heart attack, that is myocardial infarction. Fourth enzyme is alkaline phosphatase, used to diagnoses the rickets and obstructive jaundice. Fifth enzyme is acid phosphatase, used to diagnoses the cancer of prostaglandin. Sixth enzyme is lactate dehydrogenase, used for the diagnosis of heart attack and liver disease. The seventh enzyme is creatine phosphokinase, used for the diagnosis of myocardial infarction and used as an early marker for myocardial infarction. Number 8 enzyme is aldolase, used for the diagnosis of muscular dystrophy. The ninth enzyme is 5' nucleotidase, used for the diagnosis of hepatitis. And 10th and last enzyme is alpha glutamyl transpeptidase, used for the diagnosis of alcoholism. Now the last topic of the enzyme is isoenzyme. When the same reaction is catalyzed by two or more enzymes, it is known as isoenzyme. These isoenzymes are differ in chemical and physical property, which include Km value, Vmax value, optimum pH, and etc. Now, firstly, we are studying the isoenzyme of lactate dehydrogenase. The systemic name of LDH is L-lactate NAD oxidoreductase. This isoenzyme the catalyzes the interconversion of lactate to pyruvate or pyruvate to lactate. The reaction of the interconversion are given below in the PDF. There are 5 different type of lactate dehydrogenase, that is LDH1, LDH2, LDH3, LDH4 and LDH5. All the 5 isoenzymes are separated by electrophoresis. First isoenzyme, that is LDH1, is the most positive and fastest in electrophoresis, while LDH3 is a slowest one. The structure of LDH enzyme consist of 4 polypeptide chain. They consist of 2 subunit, that is M subunit and H subunit. M stand for muscles and H stand for heart, where M subunit is basic in nature and H that is heart subunit is acidic in nature. Now we can discuss their detailed structure in the table. Now in the table, the isoenzyme, their subunit constitution and principal tissue of origin are given. The first LDH1 consists of 4 subunit of H and origin is heart and RBC. The second is LDH2 consists of 3 H3M. The structure are given in the table. It has origin of heart and RBC. The third, that is LDH3, has H2M2. The principal tissue of origin is brain and kidney. The fourth is LDH4, consists of HM3 and origin of tissue is liver and skeletal muscle. The fifth isoenzyme is LDH5, consists of all 4 muscular subunit and origin of tissue is skeletal muscle and liver. The next is diagnostic importance of LDH. This isoenzyme is important in the diagnosis of heart and liver related disease. In the normal healthy individual, the LDH2 is larger than LDH1 in serum, but in myocardial condition, the LDH1 is greater than LDH2. Also, increase in the activity of LDH5 in serum indicate the liver disease. Now the second isoenzyme is creatine phosphokinase. This creatine phosphokinase help in the interconversion of phosphocreatine to the creatine using ATP. This phosphokinase present in 3 different forms, and each isoenzyme is a dimer composed of 2 subunits. Where in normal individual that CPK2 is present less than 2%, but the patient suffering for myocardial infarction, it has increased in the CPK2 value up to 20%. Now the type of CPK enzyme and their subunits and tissue of origin are given in a table. That is, CPK1 consists of 2 subunits, that is both are brain, origin is brain. Second is CPK2, it consists of muscle and brain, tissue of origin is heart. And third is CPK3, consists of 2 subunits, that is both are muscular and tissue of origin is skeletal muscle. The third isoenzyme is alkaline phosphate, that is ALP. The ALP is a monomer and the isomer is due to the difference in a carbohydrate contents. The most important ALP, that is isoenzyme, are alpha 1 ALP, alpha 2 heat liable ALP, alpha 2 heat stable ALP, pre beta ALP and gamma ALP, and so on. Where increase in the concentration of alpha 2 heat liable ALP suggests the hepatitis, and increase in a pre beta ALP indicate the bone disease. The fourth isoenzyme is alcohol dehydrogenase. The alcohol dehydrogenase, that is ADH, has 2 heterodimer isoenzymes. That is, among the American and Europeans, alpha beta 1 isoenzyme is predominant, whereas in Japanese and Chinese, alpha beta 2 is mostly present. The isomer alpha beta 2 are more rapidly convert alcohol into acetaldehyde. The accumulation of acetaldehyde associated with a tachycardia, which means increase in heart rate. It is believed that the Japanese and Chinese have increased the sensitivity to alcohol due to the presence of alpha beta 2 isoenzyme of ADH. Here we can complete the fifth unit of biochemistry, that is enzyme, and the link of the PDF is given in a description box. If you like the video, then like and subscribe and share the video. Thank you.